The human ear is made up of three parts: the outer, middle, and inner ear. The outer ear consists of the pinna, which serves as protection for the delicate inner parts of the ear, and the external auditory meatus, the ear canal, which collects sound and directs it towards the eardrum. The shape of the pinna also helps to detect where sounds are coming from in the vertical plane, that is, from above or below the ear. The middle ear is an air-filled cavity located in the temporal area of the skull. It consists of the eardrum, the tympanic membrane, the ossicles, and the eustachian tube. The tympanic membrane is concave with an area of between 60 and 90 millimeters and an average thickness of just 7/100ths of a millimeter, and it has a very rich blood supply. Sound waves enter the ear canal and strike the tympanic membrane, setting it in motion. The louder the sound, the more the membrane moves. The middle ear also contains three small bones, known as the ossicles. These are connected in a chain across the middle ear, from the tympanic membrane to the entrance of the cochlea, called the oval window. The ossicles are called the malleus, the hammer, the incus, the anvil, and the stapes, the stirrup. The handle of the malleus is attached to the tympanic membrane, and the base of the stapes, known as the stapes footplate, sits loosely in the oval window of the cochlea, held in place by the annular ligament. The tympanic membrane vibrates in response to sound. The eardrum moves the malleus, which in turn moves the incus, and then the stapes, the footplate of which moves in and out of the oval window and transfers the energy to the endolymph fluid in the cochlea. The inner ear has two distinct portions: the vestibular portion, concerned with balance, and the auditory portion, the cochlea, which is concerned with hearing. The vestibular portion is made up of three semicircular canals. Each canal has a swelling at one end, the ampulla, and two membranous sacs, the utricle and the saccule. These contain fluid and sensory cells, which move in response to head movements and travel to indicate the body's status. The vestibular system works in conjunction with the eyes and the receptor cells in the joints of the body to continuously maintain our balance. The cochlea appears as a coiled tube in the shape of a snail shell, with between 2.5 and 2 3/4 turns. It is wrapped around the acoustic portion of the auditory nerve. If we were to unravel the cochlea, it would be about 35 to 40 millimeters long. It is split into three distinct sections, known as the scala vestibuli, scala media, and scala tympani. The scala vestibuli and the scala tympani contain perilymph fluid. The scala media contains endolymph fluid. The Reissner's membrane separates the scala vestibuli from the scala media, and the basilar membrane
Surely you know the five senses: sight, smell, taste, sound, and touch. But do you know how they actually work? How do these sense organs pick up information from the surroundings and transmit it to our brains? All of the senses have something in common: sensory receptors. Sensory receptors detect changes in the environment. We'll look at 4 of the specialized receptor cell types: mechanoreceptors, chemoreceptors, thermoreceptors, and photoreceptors. Mechanoreceptors respond to forces and movements. Chemoreceptors respond to chemical substances. Thermoreceptors respond to heat, and photoreceptors respond to light. Now let's see where these sensory receptors are put to work in our senses. Olfaction is our sense of smell. The olfactory bulbs of the brain are located on the other side of the skull bone from the nasal passage. When molecules enter our nasal passage, let's say a molecule of vanilla, the vanilla molecule will bind to an olfactory receptor in the nasal epithelium. The olfactory receptor is a type of chemoreceptor because it's activated by specific chemicals or groups of chemicals. The vanilla olfactory receptor is specific to just the vanilla molecule. Once activated, the olfactory receptor sends a signal through neurons to the olfactory bulb in the brain, and then it's sent to be processed in the olfactory cortex. Humans have about 400 different odorant receptors, which allows us to identify around 1 trillion different odors. But other animals like mice have 1,000 different receptors, and by comparison, humans don't have a very sensitive sense of smell. Aquatic animals can also smell dissolved chemicals in water, just as we smell dissolved chemicals in the air. Gustation is our sense of taste. Within the taste buds on our tongues are many gustatory receptor cells with hairs that will bind to the molecules in food and send messages to our brains. Gustatory receptors, like olfactory receptors, are another type of chemoreceptor. Our sense of sight uses rods and cones as the main sensory receptor cells. Rods and cones are located in the retina of the eye, and they're types of photoreceptors. The rods allow you to see in dim light, and the cones detect color and bright light. Our sense of hearing uses complicated structures in the ear. Beyond an almost Rube Goldberg maze of membranes and tiny bones is a snail-like region called the cochlea. Deep in the cochlea are tiny hair cells, which are mechanoreceptors that register movement that's amplified from sound waves that enter the ear. Mechanoreceptors in the semicircular canals are also used to help with our sense of balance. In the skin are many types of receptors, including Merkel cells, which are mechanoreceptors that detect touch, and thermoreceptors, which can sense hot and cold. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe, and follow me on Twitter @SciencePet.
Ouch! See how quickly I moved my hand from the hot stove? Our bodies have a system in place which enables us to react really quickly, called reflex reactions. Reflex reactions are immediate, unconscious responses to a stimulus that provides us with protection and facilitates our survival. From our videos on the nervous system and nerve cells, you should already know about the stimulus, receptor, sensory neuron, central nervous system, motor neuron, effector pathway, and how sensory neurons, motor neurons, and relay neurons differ from one another. In this video, we are going to look at what happens in reflex reactions. Touch a boiling plate, face to face with a lion, eye reacting to bright light, you want a fast reaction. When your safety demands a quick response, the signals may bypass the brain and be acted upon as soon as they reach the spinal cord. These shorter pathways are called reflex arcs. Reflex arcs are built-in or innate behaviors, and we all behave in the same way. Even though the brain is bypassed for the immediate response, the nerve message is still passed on to the brain so that you can think about whether any further action is needed. So the pathway has been cut down by speeding up the central nervous system aspect. The message reaches the spinal cord and heads straight back out down the motor neuron to produce a response. See what happens when you touch a hot object. There are two types of reflex. Simple reflexes, like we have seen already, where the brain is not aware of the initial response. This aids your survival. You can also have conditioned reflexes. These involve prior thought or learning, and your body responds subconsciously because of this. Pavlov's dogs is an example of conditioned learning. During his experiments, Pavlov rang a bell immediately before feeding the dogs. He found that after a while, the dogs would produce saliva just on hearing the bell, without food being provided. Pavlov called this a learned response, which does not involve subconscious thought and is a conditioned reflex. So from this video, you should know how reflex reactions work and how they protect the body from damage. Everyone does them, and have no conscious control of what is happening. But your brain can then choose to override the initial response or act further, because this message does get sent onto the brain as well.